Getting to root causes, cellular signaling and process pathways. Why this matters. At the heart of nearly every chronic condition, whether neurological, immunological, or metabolic, is a breakdown in cellular communication. Two of the most overlooked players in this breakdown: G proteins, the molecular switches that control how cells respond to signals. We discussed G protein in the first video. Let's focus on the second. Glutathione, the master antioxidant that protects cells from oxidative stress and supports detox. What glutathione depletion disrupts? Oxidative stress defense. Glutathione is the body's primary antioxidant, neutralizing free radicals. Without it, cells suffer oxidative damage, leading to DNA mutations, protein misfolding, and lipid peroxidation. Detoxification pathways. Glutathione binds to toxins, heavy metals, and xenobiotics to make them water-soluble for excretion. Depletion leads to toxin accumulation, especially in the liver, kidneys, and brain. Immune system function. It regulates cytokine balance and supports T-cell and macrophage activity. Low levels impair immune surveillance and increase susceptibility to infections and autoimmunity. Mitochondrial health. Glutathione protects mitochondria from oxidative damage and maintains ATP production. Depletion causes energy deficits, fatigue, and increased apoptosis. Cellular repair and longevity. It's essential for DNA repair, protein synthesis, and maintaining cell membrane integrity. Chronic deficiency accelerates aging, neurodegeneration, and degenerative diseases. Drug and treatment tolerance. Glutathione buffers the oxidative impact of chemotherapy, radiation, and pharmaceuticals. Low levels worsen side effects and reduce treatment efficacy. Neurological impact. Depletion is linked to Parkinson's, Alzheimer's, autism spectrum disorders, and seizures. It affects glial cell function, neurotransmitter balance, and blood-brain barrier integrity. The parallel with G proteins. Both glutathione and G proteins are signal integrators. Glutathione modulates redox-sensitive signaling pathways. G proteins relay external messages into cellular responses. When either is depleted or dysregulated, the cell loses its ability to adapt, defend, and repair, setting the stage for chronic disease. What can we do? Referencing a pivotal and controversial hypothesis from Dr. Mary Megson, a developmental pediatrician who proposed that natural, lipid-soluble vitamin A, specifically the cis form, may help restore G protein signaling in children with autism spectrum disorders. She theorized that autism may be linked to a G-alpha protein defect, which disrupts retinoid receptor signaling in the brain. According to her research, pertussis toxin, from the DPT vaccine, may separate G-alpha proteins from retinoid receptors in genetically susceptible children. This disruption could impair vision, sensory integration, language processing, and attention. Hallmarks of autism spectrum disorders. Role of natural vitamin A. Dr. Megson suggested that natural cis forms of vitamin A, like those found in cod liver oil, may reconnect retinoid receptors to G proteins. This reconnection could restore signal transduction in the hippocampus and other brain regions. She observed improvements in language, social interaction, and sensory processing in some children after supplementation. Mechanistic insight. Retinoid receptors, RAR and RXR, are nuclear receptors that regulate gene expression. G proteins help transmit signals from the cell membrane to these receptors. Vitamin A may stabilize or restore this connection, especially in children with HLA tissue types DR3, DR4, and DR5, which bind tightly to blocked retinoid receptors. Important caveats. Her theory remains controversial and not widely accepted in mainstream medicine. High-dose vitamin A can be toxic, especially in infants, so any protocol must be carefully monitored. Her work was published in Medical Hypotheses, a journal known for exploring untested ideas. Glutathione dysregulation may be the silent disruptors behind chronic inflammation, detox failure, and neurological strain. By restoring this pathways with targeted compounds, mushroom bioactives, collagen peptides, algae-derived DHA, and NAC plus selenium, we support the body's natural repair systems. This protocol may be the missing link in reversing damage, rebuilding resilience, and empowering families to heal from the inside out. The protocol doesn't claim to cure, but it supports the body's natural repair systems, which may help reverse or mitigate damage from oxidative stress immune dysregulation, and mitochondrial strain. Together, they form a synergistic system that may help reverse oxidative damage, restore cellular signaling, buffer inflammation, support recovery from environmental stressors, including vaccine-related strain. It's a root cause restoration protocol, and may be the missing link to address the silent breakdown behind so many modern health struggles.